freue mich sehr, Ihnen heute Dr. Agnes Toth-Petroczy vorzustellen, die einen Vortrag über Disordered Proteins halten wird. Sie ist mit dem Schering Young Investigator Award ausgezeichnet worden in diesem Jahr, 2025. Und ich durfte Ihren Vortrag gestern auch schon einmal hören. Dieses Young Investigator ist natürlich für uns eine merkwürdige Bezeichnung. Sie ist Forschungsgruppenleiterin in Dresden beim Max-Planck-Institut für Molekulargenetik und für Zellbiologie, ja. Also das lesen Sie oben, Zellbiologie und Genetik. Und wenn man Forschungsgruppenleiterin ist, dann ist man natürlich keine junge Wissenschaftlerin mehr, aber in den Kategorien der Wissenschaft ist sie immer noch eine Nachwuchswissenschaftlerin, obwohl sie eben schon mehrere Preise bekommen hat. Und sie freut sich auch, den Schülervortrag heute zu halten. Es ist so, dass sie ihn auf Englisch hält. Sie kommt aus Ungarn und Ungarisch ist ihre Muttersprache und die Wissenschaftssprache, in der sie sich heimspielt, das ist nun mal Englisch. Die Präsentation ist allerdings auf Deutsch und die Fragen sind in beiden Sprachen willkommen. Ja, Sie haben die Wahl, ob Sie in Englisch oder in Deutsch fragen möchten. Zur Biografie wird sicherlich nochmal Herr Max Küchenmeister was sagen, der geschäftsführende Vorstand der Schering Stiftung, die diesen namhaften Preis eben auch verleiht. Was besonders ist quasi, dass quasi von Ungarn hat sie der Weg eben auch wirklich nach Israel, an das Weizmann-Institut geführt und später natürlich in die USA, an die Harvard-Universität. Sie hat lange in den USA gearbeitet. Und was ganz bemerkenswert ist, Lilly, ihre älteste Tochter, ist heute hier. Frau Toth-Petroczy hat 4 Kinder und zeigt eben, es geht, Beruf und Wissenschaft unter einen Hut zu bekommen. Und auch da kann sie uns sicherlich vielleicht oder Ihnen einfach nochmal erzählen, wie man das genau macht. Denn natürlich braucht man ein familiäres Netzwerk, aber man braucht vielleicht einfach auch mal die Zuversicht und die Selbstverständlichkeit. Das ist heutzutage einfach in Ordnung.
Ja, guten Morgen auch von mir. Schön hier zu sein. Wir haben gestern Abend den Herrn Schering-Preis vergeben. Das ist dann der Senior-Preis für einen etablierten Professor, der schon langjährig seine eigene Arbeitsgruppe leitet und insofern ja den Schering Young Investigator erwartet. Ich vermeide das Wort Nachwuchs, weil das bist du wirklich nicht mehr, aber in die jüngere Generation noch auf den Weg, wir haben gestern gerade darüber gesprochen, hin doch vermutlich zeitnah zur wirklich vollen Professur, der steht eben noch aus und deswegen eben in den Kategorien der Wissenschaft, das ist bei diesem Publikum dann tatsächlich ein bisschen merkwürdig, gehörst du dann eben noch zu den Junior Scientist. Ja, ich wurde schon gesagt, ich bin Vorstand der Schering Stiftung. Wer kennt denn noch die Schering AG? Oh, das ist ja tatsächlich okay, das sind die Lehrer da hinten. Ja, hatte ich erwartet, denn in der Tat ist das eine ältere Geschichte. 1851 hat der Apotheker Ernst Schering hier in Berlin eine Apotheke betrieben und im Hinterhaus Chemikalien hergestellt. Dann hat er etwas gemacht, was in der Zeit wirklich revolutionär war. Er hat sich nämlich vorgenommen, Chemikalien standardisiert herzustellen. Immer nach dem gleichen Muster, immer mit den gleichen Laborroutinen, um wirklich berechenbare Chemikalien zu bekommen. Denn das waren ja Chemikalien als Apotheker, die dann in menschlichen Körpern landeten. Und dass die möglichst rein, dass die möglichst zuverlässig, dass sie möglichst gleich waren. Das war wirklich eine Revolution. Das gab es 1850 so noch nicht und das war dann so erfolgreich, dass er 1871 damit dann eben eine Firma auch unter seinem Namen gegründet hat, aus der dann später die Schering AG wurde. Und nach dem Zweiten Weltkrieg, wenn ich da die Frage gestellt hätte, in der Zeit der Trennung in West-Berlin, da hätten vermutlich alle die Hand gehoben, denn die Schering AG war tatsächlich der einzige Weltkonzern, der in West-Berlin geblieben ist nach dem Zweiten Weltkrieg. Und wirklich ein großer Konzern war im Jahr 2005 mit 25.000 Mitarbeitern und einem Umsatz von 5 Milliarden Euro im Jahr. Wirklich der einzige große Weltkonzern und nach der Wiedervereinigung auch der einzige DAX-Konzern hier in Berlin. Aber eben ich rede in der Vergangenheitsform, 2006 wurde die Schering AG von Bayer übernommen für einen Kaufpreis vermutlich, also wenn man den Aktienwert anguckt, von 17 Milliarden Euro. Wie gesagt, kein ganz kleiner Laden. Heute Bayer Pharmaceuticals nach wie vor, nach Tesla und Mercedes, das drittgrößte Industrieunternehmen in Berlin, aber eben nicht mehr unter dem Namen Schering, den wir als Stiftung nur weitertragen. Zu eine Gelegenheit hat die Schering AG gemeint, sie müsse nochmal was gesellschaftlich Gutes tun in größerem Umfang und hat mit einem größeren Geldbetrag dann eben eine Stiftung gegründet mit dem Auftrag, das zu machen, was die Schering AG auch machte, nämlich Life Sciences, Biomedizin zu fördern. Das machen wir, und auf der anderen Seite hatte die Schering AG eine große Kunstsammlung, wie das viele solche Weltkonzerne haben, sodass wir als zweiten Standbein auch zeitgenössische bildende Kunst fördern. Ja. Einer der Bereiche Wissenschaft ist eben genau Preise auszeichnen, tolle Wissenschaft werden wir gleich erfahren. Und wie sich das für so einen Wissenschaftsvortrag gehört, wird die Referentin nochmal vorgestellt. Das mache ich besonders gerne, weil ich da sehr schön an dem anknüpfen kann, weil man, glaube ich, ganz viel da auch lernen kann, was so eine Wissenschaftskarriere auch auszeichnet. Agnes stammt aus Ungarn und hat zunächst Chemie studiert oder hat Chemie studiert in Ungarn, ist dann nach Israel gegangen für ihre Doktorarbeit und dann nach Harvard zum Postdoc. Da hat sie dann in der biologischen Chemie gearbeitet, zunehmend molekularbiologisch gearbeitet. Und dann ist sie an die Uniklinik in Harvard gegangen und hat dann Bioinformatik gemacht, also Genomanalysen, Genomik und dann eben ganz viel mit informatischen Methoden. Da wird sie sicher heute auch drüber reden, dann wirklich eben biomedizinisch und wirklich auch klinische Testungen sehr, sehr eng mit der Klinik gearbeitet. Und ich glaube, das ist so das Erste. Wenn wir in der Schule und im Studium immer noch sehr in Biologie, Chemie, Physik denken, draußen steht es groß auch an der Schule dran, so in den Fächer denken. Also die spannende Wissenschaft sind genau diese Schnittstellen und die spannenden, preiswürdigen Wissenschaftler nehmen genau diese Schnittstellen dann bedienen und als Chemikerin in der Biologie mit Genetikmethoden, mit Bioinformatikmethoden dann neue Forschungsgebiete erschließen und das dann eben auch mit einer Mobilität. Gestern mit deiner Tochter gesprochen, wie vielsprachig die aufgewachsen ist, Ungarn, Israel, USA, jetzt in Deutschland. Wir sind froh zu hören, dass du vermutlich in Deutschland bleiben wirst. Jetzt muss ich noch eine Seite sortieren. Ja, also das, glaube ich, ist eben wirklich das, was man nehmen kann. Die Fächervielfalt, das Internationale und das Dritte, das wurde eben auch schon angesprochen, möchte ich gerade den jungen Frauen unter Ihnen sagen, ja, Wissenschaftskarriere ist herausfordernd, macht Arbeit, sind viele Jahre, nicht nach deinen Arbeitszeiten in der Promotion gefragt, aber das war in Israel sicher auch viel Arbeit, es ist viel Arbeit, spannende, tolle Arbeit. Du kannst ein bisschen ja im Schülerforschungszentrum hier erleben und im Schülerlabor, das macht sehr, sehr viel Spaß, aber das ist kein 8-Stunden-Job, aber es ist eben dennoch kombinierbar. Du bist Mutter von 4 Kindern, ihr habt das als Familie gemeinsam hinbekommen, die ganze Mobilität und die Familie. Und manchmal wird das so dargestellt in den Medien, wenn ihr irgendwelche Wissenschaftssendungen verfolgt, als ob das sei das ein Entweder-Oder und entweder man ist Wissenschaftler und dann ist man nichts anderes und dann ist man so ein Nerd im weißen Kittel, so mit Einstein-Frisur. Das geht anders, das sehen wir hier heute und bitte lasst euch da nicht abschrecken, wenn ihr das überlegt. Es ist ein tolles, es ist ein soziales, es ist ein interaktives Arbeiten in die Wissenschaft und es ist absolut verbindbar mit einem Privatleben, mit Freunden, mit Familie, mit Freizeit und Hobbys. Schön, dass du da bist. The floor is yours.
Guten Morgen, good morning everyone. It's very nice to see so many of you. I'm very excited to be here. And thank you for the introduction. So I'm really happy to be here and tell you about my research in the Max Planck Institute of Molecular Cell Biology and Genetics. But first I want to start with introduction, a little bit about why I'm a scientist. So actually, originally I didn't want to be a biologist or computational person, I wanted to be an astronaut. But then I figured out that probably I wasn't born in the right country for that, and maybe I'm not strong enough for that. So then I thought, okay, then I will be a scientist. And I was already very early on interested in programming and coding. And at the beginning, I just used it to do art and reproduce my favorite artists that did these geometric art patterns. And then later, I had really great teachers in high school, biology and chemistry. And I got interested in genetics. And the wonder of why we are all so different, and it's all encoded in our DNA. And later I decided to study chemistry, and I realized that actually what I really interested deeply is proteins. So proteins are, in my opinion, the coolest biomolecules. But of course, it's subjective. And at the end, I specialized in computational methods that you will hear also today. And I ended up doing not so many experiments, but rather working on the computer, which is actually also that allowed me to combine research and family, because computational work you can do anywhere in the world, also at home, and that's made it easier to combine work and family. So I want to ask you, what do you want to actually go into? There are all these different fields of sciences: physics, mathematics, biology, chemistry, geology. So if you want, you could take your cell phones, and I have a poll on the next slide, and you could just vote so I have an overview. So there is the QR code that you can scan, and then you can vote if you want to be, or you want to study more deeply biology, chemistry, physics, mathematics, computer science, engineering, or something else, right? It's not easy to decide. I remember the time when I had to think about this at the last grade, and I really had a hard time how to decide between biology, chemistry. I was even thinking about medicine. So it's clear that it's biology and chemistry mainly that is fitting to this wonderful school that you are in. I would close the poll now and just tell you that at the end, it doesn't really matter what you choose, because all of these fields are necessary for science. And here I'm showing you my group, and you can see that some of them studied computer science or mathematics, biology, chemistry, and I even had someone who studied economy, but then later decided that she's actually interested more in biology and statistics and joined our lab. So at the end, actually this diversity is what we need in science, and we try to combine the different fields, and this is the way how we can answer actually big questions. So don't worry if you don't know exactly what you want to be, or if you change your mind later. So I would like to give you the 5 reasons that I decided, or I like, or I think it's great to be a scientist. So first, you can make an impact on the world, on a topic or a field that you like. It can be the environment, or human health, or engineering. The second reason is that you can learn how to think critically. And this is really important nowadays with a lot of fake news and AI-generated videos. How do you distinguish the truth from actually some fake information? And I have, again, a game. So as a scientist, you will learn the scientific method. The scientific method means that you learn a technique how to actually find proofs for certain questions. And here my question is to you. Do you think that the fields A and B on this checkerboard are the same color? So look at it carefully and think about it if they are the same or not. And then the poll will start. Yes, I also said yes first when I looked at it. Sorry, I said no first when I looked at it, but let's look at it again and try to prove. So A and B have exactly the same color, but our eyes, because they know that there is usually this chessboard, they try to fill in the information, and their brain is tricking us. So actually those 2 fields are the same, and if we connect them with a line, I hope you see it in these lighting settings, then you can see that they are actually exactly the same color. So the experiment, what we did here is that we connected these 2 fields, and it showed us that they actually have the same color. So this was just an illustration of a very simple experiment. Of course, we design much more complicated experiments in the lab, but this is something you can use many times when you are not sure about an information. And the third reason, as there are many questions that we don't know the answer, and we will not know it without actually someone looking for an answer, doing an experiment. And if you ask ChatGPT, often it will pretend that we know the answer, but many times it's not the case. So we need scientists. The third reason is that this is a job when you can still remain a child, even as an adult, and you can keep your curiosity and your interest for subjects in the world that you care about. And the fifth reason is that scientists are actually not on their own. This is something we do as a team, and this is a lot of fun. So here I'm again showing you some pictures from my lab on our free time when we do some activities. But also I want to show you that in my group, These are all the nationalities that I had so far. I'm 7 years now a group leader, and you can see that it's a very international group, and it's a wonderful environment to work together. So with this introduction now, I would like to switch to some more serious topics, and I would like to talk to you about my research. And I would like to illustrate to you that biological systems are very, very complex. They are much more complex than what humans can build. And I would like to show this to you by illustrate this to you with some numbers. So the human body is actually built from 30 trillion cells. This is more, 300 times more than the stars in the Milky Way. So the human body's complexity is incredibly huge. How can we study such a complex system? Some people decide to study some organs or different cell types, neurons. I decided, and I'm most interested in proteins. And there are 20,000 different proteins, for example, in human. Many of these proteins are essential, so if they don't function well, they can cause a disease. So let's look at it, how proteins are produced in the cell. Probably you have learned it already in biology class, that the DNA encodes all the information to produce a protein. It's like a cookbook that has the recipes to produce proteins. These recipes are the genes. And then these are transcribed first to the RNA and then translated by the ribosomes that builds the proteins. And the proteins are the machines in the cell that do all the work. They are really like machines. However, in this information processing from DNA to proteins, there are errors happening. We call these mutations. And these mutations mean that there will be changes in the sequence of the protein. The protein sequence has a long chain of amino acids, then folds into these beautiful 3D shapes like you see here, this pink color. So if these mutations happen, are these always bad? So many times when a mutation happens, that destroys the protein, it loses its function, and that can cause disease. However, several mutations can be neutral or actually change the protein in a positive way. And this diversity generated by the mutation is the driving force of evolution that actually helps species to adapt to changes. So my question is that we would like to answer in my lab, how can we understand which mutations are good or bad and how they impact the protein's function? So let's look again what proteins look like. So if we just think about the sequence, it's a sequence of 20 amino acids. These are the letters. If you would play Scrabble with these 20 letters, you could basically build proteins yourself. But these are much longer than the words we usually build. They can be hundreds or even thousands of letters long. And if there is a mistake in them, that could be a deletion or a typo, for example, that you would make. And then these long amino acid chains fold into some three-dimensional structure that is often very, very specific. In this case, we call this protein the beta barrel. It looks like a barrel. And this is a very famous protein, a fluorescent protein, that allows us to see proteins inside cells because it's fluorescence. Maybe you have seen it already in the lab. And this concept that proteins have a very file-defined 3D shape is a so-called Schlüssel- und Schloss-Prinzip, lock and key principle, which means that a protein has a very specific shape that fits, for example, a ligand, a drug, or another molecule that it has to bind, and that's why it has this very specific shape. This is kind of the textbook knowledge. Maybe you have also heard about it. However, in the last years, there were some breakthroughs, and this is something that probably you haven't seen in textbooks yet. This is new research from the last 25 years, where people actually realized that not all proteins have these beautiful 3D shapes, but there are so-called disordered proteins, intrinsically disordered proteins, or regions in proteins that don't have these beautiful structures. So this would be here the structured part, and this would be an unstructured part that is flexible. However, it contains 33% of the human proteins. And the next breakthrough that I want to mention is the discovery of so-called biomolecular condensates. These are protein communities that form in the cell, kind of spontaneously. You can see these little droplets they look like liquid droplets, or when you mix oil and vinegar, you have these little droplets. It's exactly the same process, like phase separation, liquid-liquid phase separation, how these are formed. I will tell you more about later. And the third revolution that I want to mention is the AI revolution. We have now ChatGPT, we have self-driving cars. Of course, all this technology can be also applied in biomedicine or biological studies. And interestingly, because we have been studying disordered proteins for decades, we have a lot of information on them. So now we can teach an AI algorithm how to recognize them and predict from the sequence of a protein what it would look like. And the Nobel Prize was actually given for this AI tool that's called AlphaFold, developed by Google DeepMind, which is amazing. However, for the disordered proteins, because also we didn't have enough data on them, this algorithm doesn't work. And here it predicts this nice structure. The blue part is the ordered protein, and the disordered one around is like a hairball-like structure that doesn't help us to understand its function. So disordered proteins are flexible, as I said. They move around, they are very plastic, they can bind many partners. So why would we need such proteins in the cell? I would like to illustrate one function that is easy to see. First, just my analogy. So just that you remember, ordered proteins is like pasta, and disordered proteins is like cooked spaghetti that is very soft. And as pasta needs specific shapes, it has different tastes. It contributes to its function, while spaghetti has to be flexible, which is an analogy for disordered proteins. So why would you need such disordered proteins? One example is the muscle. So in our muscles, when we extend our arms, for example, and close it, the muscles have to move, contract, and expand. That means also that proteins have to contract and expand inside as well. And there is a protein called titin, that's the longest protein in the human body. It's 36,000 amino acids long. This is here shown in yellow on the side. So this protein, when we extend our muscles, then also completely extends. And when we contract our muscles, then the protein folds. So this is an example where you can see that being flexible and like a spring-like molecule can be very, very useful. And what happens if this protein has some mutations that are bad? It can cause, for example, heart disease, when the heart muscle cannot contract correctly anymore. So how can we identify which mutations would hurt this protein, or in general proteins? So one method is using information from evolution and looking at proteins from other organisms. For example, in this case, we are looking for the titin protein in different species, and then we find the gene in dogs, in whales, in chicken. That makes sense. All of them have muscles, and we can look at those proteins. In trees, we don't find them. Makes sense again? Trees don't have muscles. And then we can compare the sequences of those proteins from the different organisms. Usually we do this with actually hundreds of different species. I'm just showing you a small example. And to illustrate what we are looking for, I just take a snapshot of a small region of the sequence. So this is a protein sequence from 3 different species. You can see that there is a part of it that is important, that we call evolutionary conserved, because although these species have been diverged for 300 million years, and there were a lot of mistakes when the DNA was copied, still this region is exactly the same between chicken and dogs. However, this other region is not conserved. It can change, and this is actually a way how diversity is evolving, that we have all different hair colors or eye colors. These are not important regions. So the functional regions would be the conserved ones, and the variable regions could be the non-conserved ones. And if a mutation happens in a human, for example, in an important region that is functional, that could cause disease. If the mutation happens in a non-conserved region, that's usually fine. That doesn't cause disease. So using this trick and information from other organisms, we can define and design tools and algorithms that take the sequence of a protein, also structural information, look at the mutation, compare it to other species, and then train an algorithm that at the end decides, is this mutation bad or good, would cause a disease or not. So in my lab, we also developed such a tool, and we decided to focus on so-called actionable genes, medically actionable genes. What does that mean? That means that for this disease, there is actually some treatment or preventive diagnostics that can be done if someone has a bad mutation in such a gene. But often we don't know if the mutation is bad or good, and that can cause anxiety if you have a mutation, but you don't know if it could cause a disease. So it's really important that we can distinguish them. And maybe you remember the story of Angelina Jolie, who decided to test herself because there was many breast cancer in her family. And it turned out that she carries a mutation, a bad one, and then decided to preventively do some surgery and prevent to have breast cancer later. And actually, 3% of the people have a risk to carry a mutation in one of these genes that are actionable. So we developed this tool called DMAG, Deciphering Mutations in Actionable Genes, that it has relatively high accuracy to decide if your mutation would be bad or good. But unfortunately, the algorithm doesn't work very well for disordered proteins, as I would like to illustrate why. So there are two reasons. Disordered proteins and disordered regions are actually new, relatively, evolutionary speaking. So they are much more in eukaryotes and mammals and humans especially than in simpler organisms where we have a lot of sequencing data available. Another reason why it's difficult to study them because they change a lot. They tolerate a lot of mutations. So if we compare these sequences from an ordered region, it looks very pretty. You can see where are the conserved sites and the non-conserved sites. While in the disordered one, it's a mess, and it's not so easy to use these algorithms or kind of methodologies. So we decided with my first PhD student that we would like to solve this problem, that we don't know how to look for these disordered regions. And we developed an algorithm that is called SHARC, that stands for Similarity Homology. There was another slide, no? No. Okay. Similarity Homology Assessment by Relating K-mers. And my PhD student really loves sharks, so he had to come up with an acronym for this abbreviation. But basically what this algorithm is doing is that it's comparing not the sequence, but breaks it down to small words, and we compare those words. And it's kind of like breaking down this fish as an analogy for proteins into what they have, like fins and eyes and colors, and then look for similarities, like these all have stripes, or these other fish are all yellow, and then identifies basically similarities that are not encoded just in the sequence. And capture is a version of the tool that can identify exactly which regions are functional. And then we can use this tool, found that this is the bit that is functional. It's just a 4 amino acid long small motif. And when we mutate this motif in the lab and we change it to all alanines or delete it or shuffle the sequence, all of these changes will lead to reduced activity of this protein that we measure in the lab, which kind of proves that this is a functional region. And we can do this now because this is a computational algorithm to any protein and study any organism or anything that we are interested in. So I would like to now break a little bit the flow because we will switch a bit topics and ask you, what do you think? How many proteins are in a mammalian cell? 20,000, 1 million, or 1 billion, which in German is Milliarde, which is a bit confusing. It's nice that so many of you are voting. It's tight, but the correct answer is actually 1 billion, so 1 Milliarde, so the highest number, basically. It's incredible. So there are so many proteins in a cell. There are 1 to 3 billion proteins in one cell. So it's a fascinating question. How do these proteins find each other? How do these proteins find the right partner who they have to bind, what they have to function together? So somehow proteins have to organize. It's not just a mix. It's not just a soup inside a cell. So this is a fascinating question. And one answer that probably you have learned in school is that there are organelles. There are mitochondria, there are lysosomes, there are endometric reticulum. Probably you learned about these organelles. But the new research, what I already mentioned to you, showed that there are also membraneless organelles that we call biomolecular condensates. And these are protein communities that assemble together because of similar properties and function, like AGs in the school. When you have for Fußball AG or the Musik AG, the proteins have to find basically their specific communities, and these are self-assembled and they don't need a membrane. Here I'm showing you a fluorescent image when people label two different proteins with some fluorescent dye, like the protein I showed you before, and you can see how they form and actually they fuse. So they really resemble, like when I mentioned the oil and vinegar, when you mix balsamic vinegar with oil, for example. And that's exactly the process that researchers identified as phase separation, liquid-liquid phase separation. And you can see in the cell this liquid-like little droplets forming. So my lab is interested in what is inside these droplets, so which proteins are in each community, how do they know where they belong, and also how did these communities evolve. So these are the questions that we are interested in. And again, we use computational methods. But since this is such a new field, it's really just the last 15 years that we know that such kind of condensate exists. There's not really enough data yet, but there's a lot of publications, scientific publications. So we decided in my group that we will collect the information. We also use AI tools. We use ChatGPT-like algorithms as well to collect the information. But at the end, we have to standardize and also manually curate this and build a standardized database that then we can use to develop a computational algorithm that can predict behaviors and then test it in the lab. So the input to such AI algorithms is always first the experimental data. So we built this database. We call it CD code. That stands for Condensate Database and Encyclopedia. And we collected condensates from different organisms, their components, drugs that can impact them, and also diseases that are linked to such condensate functions. And we released this now to the community. We have now the second version actually already online. And we used ourselves this to develop another computational algorithm that takes again as an input the protein structure and sequence, and then predicts if a protein would form a condensate or not. And our algorithm is called PICNIC, proteins involved in cells, in condensates in cells. Now we can use these algorithms to look across species, and we were interested to find that they are everywhere. They are also in archaea and bacteria, and even viruses have condensates when they infect us. So why do we care about condensates? Also because they are important in disease. So if many diseases are linked to malfunction in condensate formation. There are too many condensates, or they are at the wrong place, or they have different properties. And here I'm listing you a few examples, for example, in cancer, neurodegeneration, but also infection. As I mentioned, viruses form condensates when they infect our cells, and they assemble inside these liquid-like droplets, the new viral particles, for example. So the next question that we would like to understand how mutations impact condensates. And there is a famous disease, maybe you have heard about it, it's ALS. That's a neurodegenerative disease that Stephen Hawking had. And it's linked to a protein that is disordered. It's called FAS. And just a single mutation in this protein can cause a disease. So researchers have looked at it. What happens to the condensate formation of this protein in cells? And they saw that the normal protein, the wild type, forms condensates, and also the mutant forms condensates. And there was no difference. But then when they waited a little bit, which is also what happens in this disease because it's usually a late onset, then they saw that there are these spike-like, star-like structures that are forming. The condensate hardens and grows these fibers inside. And it looks like in 3D like a Christmas star. So researchers now hypothesize that these changes in the condensate formation are linked to the disease. And this can be a new way how we can target several diseases, not just the protein as a drug target, but actually targeting the condensation, that the protein, for example, doesn't form these fibers anymore. So my lab is now focusing on trying to identify which mutations would influence condensate formation. And with that, I reached the end of the presentation. I would like to thank my institute, which is the Max Planck Institute in Dresden. Here I listed some of the tools that we have developed over the years. And most importantly, I would like to thank my wonderful lab. This is the current members and the past lab members, my collaborators, and the funding sources. And also, I would like to thank my teachers, because I wouldn't be here if I didn't have a wonderful chemistry and biology teacher and a math teacher that happened to be my mom. And also my mentors along the ways, my master's supervisor in Hungary, where I actually started to work with disordered proteins. My PhD advisor, Dan Tawfik, whom I worked on protein evolution. And then Debbie Marks at Harvard, who I learned the computational algorithms and statistics. So you can see how you could combine kind of the path, like what I learned from my mentors, to build up my own research program. And my current mentor, Antoni Hyman, who is also the pioneer of this condensate research in Dresden. And I also want to mention that I'm really, really grateful for my mom, who supports me and, for example, comes to me to conferences when I have babies who I don't want to leave at home. They still need to breastfeed, for example. And this is a picture that I took when we went to a conference in Heidelberg together. And these are my kids and also my husband, who is also a scientist. And if you want and still have the energy, we prepared a very short video with my lab. It's 2 minutes. That could be like a summary, what I said, with German subtitles. So I would play that. I'm Agnes Toth-Petroczy. I'm a research group leader at the Max Planck Institute of Molecular Cell Biology and Genetics and the Center for Systems Biology in Dresden. In my group, we are fascinated by proteins that are the most exciting and important biomolecules in the cell and their evolution. We would like to specifically understand how the sequence of the proteins determines their three-dimensional shape and function. And we specifically focus on the types of proteins that don't have a well-defined 3D shape, but are flexible, so-called disordered proteins, and these are much more difficult to study both computationally and experimentally. Many diseases are actually linked to protein dysfunction. It takes only a single change in the DNA to alter the protein sequence and cause a disease. And such mutations can happen not only at the DNA, but also in the process of producing those proteins that we are also interested in.
And I use mass spectrometry and computational modeling to detect errors that cells make when they produce proteins, because these errors are important both for protein evolution as well as that they contribute to diseases.
And as everything in life, these mutations can have a bad or a good effect. They can cause disease, but also this variation in protein sequences is a driving force of molecular evolution. Proteins are not on their own in the cell. They bind other proteins or other biomolecules, and they form molecular collectives. We call them biomolecular condensates. We would like to understand what determines in the protein sequence which protein belongs to which biomolecular condensate. There are billions of genomes from different organisms sequenced now, and we can use this big data, this information, to develop machine learning models.
My background in mathematics and computer science allows me to create machine learning algorithms to predict the properties of the proteins.
I'm excited to work with an interdisciplinary team of scientists. We can answer questions from many different angles. We develop software, databases, tools for the scientific community, and at the same time we also perform computational experiments and wet lab experiments, for example using mass spectrometry, microscopy, and sequencing. I thank you for your attention, and I'm very happy to take questions. You can ask in German or in English, it doesn't matter. I can also try to answer in German. So please ask. Thank you. That's a very good question. it depends where we look, right? So we would have to do sequencing as well as looking at the protein sequence as well, which we can do, for example, with mass spectrometry. That's one technique, when we can see what happens at the protein sequence exactly. I answer in English, yes. Yeah, thank you for the question. We actually, our main focus is now these condensates, which is very, very exciting. And exactly what you said, to understand which mutations would impact the condensates, that mutations that are linked to disease, then what happens to these condensates. And it's really interesting that in neurodegenerative diseases like this ALS, these cells, these neurons are really long and very also old, so they can be like 1 meter long, and there's differences along the cells, for example. And we are now looking at if there are different condensates or different behaviors along the cell. And we also have a collaboration on viral condensates. So we work together with someone who studies viruses, and they also, as I mentioned, form condensates. There's another aspect, disease aspect so far. Yes, that's also a very good question. That's this video that I showed you at the beginning was such a simulation. So you can indeed simulate with molecular dynamics simulation these disordered proteins, and people do that. To simulate this behavior of assembling into something, for that you need so-called coarse graining, which means that you don't describe the molecules by all the atoms, but actually you say one bead is one amino acid, for example, or even more rough coarse graining. And people do that. Usually it's very good for individual systems when you want to understand one protein very deeply. For systematically, like running this on all the 20,000 proteins, that's still difficult. Yes, the complexity of the cell, I would say, is very, very high. So these models that you mentioned, they usually focus on one aspect of cell behavior. For example, maybe metabolic pathways, so this is, I think, the ultimate goal of many computational biologists is to build such a model. When we have basically an in silico copy of a cell that knows everything about it. It knows the proteins, it knows the RNAs, the lipids, the metabolites that are inside, and then you tell, if I change this, if I give sugar, or if I add a mutation, what happens to the cell? I think it's possible, but it's still probably a few years or decades. Deep learning needs a lot of data. So I think it's a combination of a lot of more experiments, but also systematic experiments, because that's something we faced also with the condensate database when we built it, that sometimes experiments are like apples to oranges. They are not the same, and you can't learn from them because there are too many changes between parameters. So if people do systematic experiments for this purpose, I think that's the first step, and then comes deep learning. Because the complexity is so high, we could say we humans will not be able to build models from first principles, from physics and chemistry. We give up and we say, let just the computer learn the complexity. Wow, that is also a very good question. You know, recent papers. Yes. Very stable form of several disordered proteins is these aggregates, these kind of fibers and like amyloid plaques, if you have heard about such things that people see in the brain of people with neurodegeneration, for example. And often these proteins are disordered when they function normally. And then when they form these very stable aggregates, they kind of switch behavior and then they become these plaques. So it is often linked to disordered proteins. Yeah, I actually a little bit simplified this. So it's not that they are disordered and ordered proteins. It's actually a continuum. And also a disordered protein can behave like an ordered one under certain conditions. So just to give you like a bit more nuanced view of this, it's it's more complex. And mutations can also switch this behavior. Yes, that's true. Indeed, with frameshift mutations, I don't know if you mean frameshift in the DNA level or when the protein is produced. These are differences. Actually, I skipped that part from this presentation, but maybe you heard it yesterday in the Schering award ceremony, that when proteins are produced, there are also mutations. So not only at the DNA this can happen, the changes, but also when the proteins are produced. And often frameshifts happen when the ribosome that generates proteins shifts and then generates a new sequence. Actually, an algorithm would not be able to find them, I think. So that would have to be developed, that it looks for potential frameshifts that could solve it. It's also a very good question. Thank you. Yes, indeed, there is a type of AI that can hallucinate. This is not the type that we used. So these are kind of generative models that generate new information. So our tools that we have used, they are so-called classifiers. These say yes or no. So they basically just decide between two options. But the AlphaFold tool that I mentioned, which predicts from a sequence if it forms, well, what type of structure it forms, the second version of it is such a generative model that can also hallucinate. And it was very interesting when people applied it on disordered regions, it hallucinated structures. So the first version was just completely useless because it said, I know proteins should have structures. So let's just pretend that these disordered regions also have structures. And then what the Google scientists did, they trained the new generative algorithm in the old one that knew that these regions don't have structures. And they used this data to kind of fix this mistake that it hallucinates ordered proteins. So it's very important with any type of AI tool that there is some validation and some real-life experimental evidence that what it predicts is actually true, because hallucination is a big problem. Yes, when we focus on these actionable genes, this is actually also the genes that have the most data. Because in the US especially, people are screened for these diseases. As I showed you, 3% have a risk to have such a disease mutation. So I think it's quite widely sequenced. Probably there is still some kind of gap. So when we assembled that data, we specifically looked for all kinds of biobanks. So there is a UK biobank, maybe you heard about. We looked at some Japanese and Korean biobank data, Estonian biobank. So we collected basically other ones that we could find. But some countries just don't have a biobank. Actually, Germany also doesn't have one. So there is some underrepresentation there. We haven't sampled specifically or tested if our algorithm works better in one population or the other. It could be. Yeah.
Is that something you're planning to do, or is this just a gap in the data? A current gap, of course.
It's actually all the matter of the amount of data, because if the data is too small, then there's nothing to do about it, unfortunately. But I think it's important to have awareness about it. And we could try just showing at least how well the algorithm works in different populations, and then highlight that there is a need to generate more data for those populations, ethnicities, where there is not enough yet to have good algorithms. So that we should do. I don't know that much about this, what you mentioned, so I don't know. But I think, again, I just have to highlight that the biggest bottleneck in many biological or biomedical AI tools is the lack of data. And indeed, depending on how you slice the data and design your training and testing set and validation set, there's a big difference in performance. So if there are more advanced ways to sample the data and kind of choose in the right way how to split it, that will help. I would give the advice that if you like the daily activity of doing science, reading papers, thinking about problems, or doing experiments, or programming, then just go for it, and then you will see if it's indeed as much fun as you imagine. Because I think it's hard to imagine what the daily life of a scientist is when you just study. So just try it, or look for an internship, look for a place that accepts high school students, or even during your studies, like some small internships, and then you can try, and don't stop if the first place is not the right one. Of course, you may not have found the right topic yet, or it may be not the right lab, maybe not the right type of supervisor. So if you think that this is what truly interests you, then don't give up, and look for your place, which can be anywhere, right? Science doesn't have boundaries and doesn't care about borders. You can try in different universities, different countries, different topics. As I said, you can move between fields. And also one of my best PhD students was a biologist who never coded a single line before he joined, and then ended up developing algorithms. So you can learn and keep learning. I remember when I looked for a postdoc position, so after the Doktorarbeit macht man so diese postdoctoral Arbeit. I sent 10 emails and I got 2 answers. So I was very disappointed. So that's how it starts. So that's why I said don't give up. There are many busy professors, but there are many places. So then I guess you just have to believe in what you do and insist and take the courage to apply or apply again. Of course, it's very important that you send these applications in a very targeted way, so that let's say the professor where you apply understands that you really want to work on this topic. It's not just that you send 100 emails with changing just the subject line, but you actually have an interest in this topic. And usually when they feel that it's something that you really care about and you're curious about, then you will get an answer. Then you get invitation, hopefully, for an interview. And then from that point, it's about the interaction with this specific research group. It wasn't an option not to manage, so I thought I would like to have kids. I had Lily, who sits here, who is now 12 years old, when I was a PhD student. So I already started very early on as a mother. And I thought if it doesn't work, I will quit science, but let's try. And it worked. I have also wonderful kids who tolerate traveling between countries or going to conferences. But I think also I learned a lot from managing the family, and it's very similar how I would have to manage a lab. So they kind of complement each other, and I learned a lot from my kids, how to ask questions in a simple way that helps my scientific thinking. So they really complement each other. I don't see them as two separate things. I think I'm a mother scientist, and I would be a worse scientist if I wasn't a mom, or a different scientist at least. I think worse. And I just have to make it happen. And of course I have to compromise. Sometimes I have to compromise on, I would like to work longer, but I can't because I have to pick up the kids. Or the other way around, that I have to skip a concert because there is a very important appointment at the same time. So I can't do everything 100%, but I think it's like that with many things in life, that you just have to define your priorities. That's a very good point. I think also that scientists should speak up, and as I mentioned, the scientific method that I try to illustrate with this funny optical illusion, it's something that should be used also in politics and also in the news, because there are many times I also think that reports are not actually critically, don't think critically enough about what they report. So there is a responsibility on scientists, and that's something that also me, as I'm getting older and more senior, I will take on, I think. At the beginning, as you say, there is publish or perish. You try to survive, you try to build your lab, you try to establish your scientific group. and get a permanent job. And then you should really give back to the society, and you should make sure that your research is truly, and your knowledge is also benefiting a wider, broader public and the country, or globally, the world. Yeah, it's a balance. So the first question was how to be a scientist without traveling, right? Or moving around too much. So that's actually something that since COVID pandemic improved, because then people realize that actually you can have a lot of meetings and a lot of interactions also online. And since then we have Zoom meetings, conferences, symposia that are broadcasted, which is also helping, for example, countries where students wouldn't just be able to travel because there is not enough funding. So it became also more inclusive in this respect. And also with computational work in my group, many people work from home 2 days a week maximum, which I think helps. But on the other hand, we also need interaction. So it's really a lot of fun when we just stand at the whiteboard and we draw ideas and we discuss and we brainstorm, and we just need this human face-to-face interaction as well. So you need both. But reducing travel is also something that I really minimize because of my family and the kids. And I give online talks sometimes. So that's another way to connect. But yeah, and I think your other question was if you can start already doing science from home. Yes, totally. So I would say also there are many online courses that can be done, and also remote work is possible. I may have had, but I may have not noticed, because I was raised by a single mom who was a math physics teacher, and I knew that women can do everything. So I wasn't sensitive to noticing issues, and I was always feeling very, very strong and very well appreciated. There is also sometimes this kind of self-fulfilled prophecy, when someone doesn't feel self-confident enough and then feels some form of negativity that may not even be fully true. So this is also something I would say to just think about. I think there were some moments when I felt that I'm not being taken serious and I had to raise my voice, and it may have been because I was a woman, when I had to kind of defend my opinion, so that happens. But I myself did not, luckily, I did not experience any form of discrimination. I also would like to thank you all for these great questions, for this interaction. I really, really enjoyed it. This is a wonderful initiative. So thank you so much for being here.